Now we begin our discussion of Chapter 5, where we're going to look at our eukaryotic cells and microorganisms. And this just starts out with a table of eukaryotic organisms that we're going to study in this course. The protozoa are the unicellular ones. A few of them are colonial. The fungi and algae, they can be unicellular, colonial, or multicellular. And then the helminths and the arthropods are multicellular, except the reproductive stages. And of course we're talking about parasitic worms and some animal vectors of diseases here. Now this is a typical eukaryotic cell that we've looked at so many times that we've studied a lot in 103. But to talk about it a little bit more, how's it organized? First of all, there is a boundary of the cell. It's got some external structures sometimes, and it's got organelles within the cytoplasm that's inside the cell membrane. The external structures are the glycocalyx, which can be either capsules or slimes. The capsules are secured more tightly. And then the boundary of the cell is the cell wall and the cytoplasmic membrane, or the cell membrane. A lot of cells don't have a cell wall, remember that. But it's the boundary of both of them is the cell wall and the cytoplasmic membrane. Now, the organelles that are within the cell membrane, of course, you've got a cytoplasmic matrix. You've got the nucleus that contains the genetic material, an endoplasmic reticulum. You've got the Golgi complex and the mitochondria, a little typo there. Also got chloroplasts, and then some organelles that function for locomotion, the flagella and the cilia, ribosomes, and the cytoskeleton. And hopefully by now you're familiar with what all of these do, but we'll be talking about that as we go through. You might need to review some of these if you need to. Now, looking at the locomotor appendages, the flagella, this is a long sheathed cylinder, and it contains microtubules, and it's covered by an extension. It has a sheath that's part of the cell membrane, and it's going to be 10% thicker than the prokaryotic flagella. And the way it can move is by whipping back and forth, or else pushing off in a snake-like fashion, where it lashes and grabs the substrate and it pulls. The cilia are very similar in structure to the flagella, but they're shorter and more numerous, and you only find them in a small group of protozoa in some of the animal cells, not many. And the reason for them again is locomotion, feeding, as well as filtering. And you can see there at the bottom in figure B the power stroke and the recovery stroke, how they help the cell to move. Now the glycocalyx is the outermost boundary, and it's what's in contact with the external environment of the cell. And it's mainly polysaccharides, and it can either be a slime layer or a capsule. Remembering that the capsule is going to be more anchored and more secure. It functions to help the cell adhere to things, for protection, and also for signal reception. And beneath the glycocalyx, there's a thick, rigid cell wall in your fungi and most of your algae. But the protozoa and some other algae and the animal cells don't have a cell wall at all. They only have the cell membrane. Now the cell wall is very rigid, and we know it's for structure and support. In fungi, it's composed of chitin or cellulose. In algae, the chemical composition varies. Most often it's cellulose or calcium carbonate. You know, we describe the cell membrane over and over and over again as a phospholipid bilayer with proteins scattered throughout. And we talk about how it's selectively permeable. It lets some things in, and it won't let other things in. It lets other things out and won't let some things out. So it picks and chooses what can move across that membrane. Our first concept check question, which part of the eukaryotic cell is responsible for signaling or contacting the outside environment and signaling between cells? Would it be the flagella, the cell wall, the glycocalyx, or the cell membrane? Think about what's in contact with the outside environment. The glycocalyx.